Hello everyone, thank you for taking the time to join our webinar. Today's topic is using NGS for CRISPR validation, antibody screening, and metagenomics. Our webinars are designed to provide helpful guidelines and information that will keep you up to date on important areas of life sciences research. After the webinar, we'll be sharing a copy of the slides and the recorded webinar. If you are signed into a Google account, please use the chat box on the right side of the screen to submit any questions you may have. If you did not register for the webinar earlier, there is a registration link in the video description. Today, we'll begin with a brief overview of challenges with traditional Sanger sequencing. Next, we'll discuss amplicon sequencing, why you might use it, and how you apply this into your own research with next-generation sequencing. Finally, we'll go over some of the workflows and analysis that we can do with Amplicon-Seq, including CRISPR validation, antibody screening, and even metagenomic sequencing. Now, I would like to take a moment to introduce my colleague, Dr. Christopher Smenjak, an NGS specialist at ABM. After completing his PhD at the University of British Columbia, where he studied stem cell regulation, he joined ABM to help scientists achieve their goals. With nearly 10 years of experience in research and experimental design, he can assist with nearly any project, from initial setup to post-sequencing and data analysis. Alongside him, you have me, Nancy Yao, a product specialist with over 7 years of R&D experience in molecular biology and biochemistry. Our main goal is to provide researchers with the support that ABM has been renowned for. For those of you who may be new to ABM, I would like to provide a bit of background on our company. ABM was founded in 2004 in Vancouver, Canada, and we have been working hard to catalyze scientific discoveries in the field of life science and drug development for 15 years. We are constantly one of the fastest growing companies in the region, and we have been working to become known as a reliable source for researchers all around the world. Due to our success, we have been able to expand our facilities, beginning with a branch in China in 2013 and a new facility in the United States in the very near future. These expansions have put us in a position to better work with each of you and to provide world-class support whenever you may be. With our team of dedicated scientists, ABM is committed to empowering researchers with the latest innovations for all of their scientific needs. Before we jump into the amplicon sequencing, let's first go over some of the challenges with Sanger sequencing and to better understand how next-generation sequencing can help to solve this. With Sanger sequencing, there are a number of advantages. Most importantly, for a lot of researchers, Sanger sequencing offers quite low cost for small sequences, and the sample preparation is generally quite simple. But there is also important to consider that this method is for low throughput, meaning it's challenging to handle a large number of samples. It requires specific validated primers for each step, and if your sample is mixed, the result will be very difficult to interpret. For instance, Sanger sequencing generally produces a trace or chromatogram with a sequencing result. You can use this on the left side. You can see this on the left side of the slide. This is then used to determine the nucleotide sequence for a given region. But what happens if you have a sample that's not one single DNA sequence or is heterozygous at some sites? In these cases, the chromatogram will be hard to read, and there may be even regions where it's impossible to tell what a nucleotide is at a particular site. In these cases, the way to determine the sequence from mixed samples is to clone the DNA fragments into vectors, isolate individual bacterial colonies that contain one unique plasmid, and then sequence each plasmid insert separately, which can take weeks or even months. Next-generation sequencing with Illumina can help avoid this step. It offers a number of advantages. First, it's possible to sequence without prior information. There are no special sequencing primers after you have generated your amplicon, and there's no cloning required. In many cases, you can directly ligate sequencing adapters to your sample and then sequence. Next, NGS is massively high throughput, allowing you to sequence thousands of different sequences at once at the same time, and also offers single nucleotide resolution, even from mixed samples. This approach is perfect if you also want to study variations between mixed samples. There are a few things to consider, including the fact that you will need access to a sequencing machine and a technician to operate it. The cost for NGS is typically higher than that of the traditional old-school sequencing. And due to the volume of data output, analysis can also be very challenging if you are not familiar with the types of data or the analysis tools that are available. While Sanger sequencing isn't well suited to sequencing mixed amplicons, NGS can address this with amplicon sequencing. With amplicon sequencing, the mixed samples are not a problem, you can easily obtain clear results, and no cloning is required. So, what is amplicon sequencing? Amplicon sequencing essentially refers to the deep sequencing of PCR products and amplicons. And it is useful when you know the target region you want to amplify, and you have amplicons that are less than 600 base pairs. And you have mixed samples or samples where you expect a lot of sequence variations. For example, if you want to sequence ribosomal RNA from mixed bacterial samples, you can do this by first extracting DNA from your samples, using primers specific to a conserved target region, and generating amplicons for the rRNA gene. This will generate hundreds of amplicons with different variable regions between your conserved target regions. These can now be sequenced with NGS to tell you about the different species present in your sample and their relative abundance. Amplicon-Seq has increased importance with the growth of Illumina platforms and emerging fields of research, including CRISPR studies where you need to validate edits that Cas9 has made in your samples, and antibody screening where it can be used to decrease the time it takes to identify unique sequences for the difficult to develop antibodies, and metagenomic studies where researchers want to study mixed populations from water, soil, or gut microbiome samples to learn about the microbial diversity. Now I would like to hand things off to Christopher, who will go over the amplicon sequencing in more detail and briefly discuss its applications, including CRISPR validation, antibody screening, and metagenomics. As a quick reminder, please post any questions you may have in the chat box during the course of the webinar for our Q&A section.
Thank you, Nancy, for that introduction to Sanger sequencing and some of the challenges that Amplicon-Seq can help address. Next, I'll do a brief introduction to next-generation sequencing with the Illumina platform. Next-generation sequencing can be used for a number of projects, including whole genome sequencing, studying changes in gene expression with RNA-seq, as we've discussed in previous webinars, or metagenomic studies from environmental samples and other applications as we'll learn today. For example, you can use amplicon sequencing if you wanted to study what types of edits might have happened in a CRISPR experiment that you set up. Or it can be used for antibody screening to determine what antibody sequences exist in your pool of antibodies, or for metagenomic studies to determine what types of organisms are present and in what proportions. All next-generation sequencing follows the same basic workflow, where you start with input material, ligate sequencing adapters, and then proceed to sequencing on one of the Illumina platforms. Next, I'll go over a couple important terms to know for all next-generation sequencing projects. The first is the read, which is a sequence of nucleotides that is sequenced. Here you can see a double-stranded DNA molecule that will be read from one molecule end to the other, and that's the sequencing read. The sequencing read length can vary, and the length refers to the number of nucleotides that would be sequenced with each read. For instance, you can have read lengths such as 75 nucleotides, 150 nucleotides, or up to 300 nucleotides. Read lengths of 300 nucleotides are the longest available reads typically available and suitable for most Amplicon-Seq projects. Next, you have single-end sequencing, where a DNA fragment is read but only from one strand. The alternative to this is paired-end sequencing, where a DNA molecule is read from both strands, giving you one paired read. Next, you'll need to know which type of sequencing strategy might be appropriate for your project. Generally, for amplicon sequencing, paired-end sequencing is the standard. This is because most amplicons are typically larger than 75 nucleotides, and with this strategy, it's possible to completely cover most amplicons. Picking the appropriate read length is important for a number of reasons. In this example here, you can see that we have an amplicon of about 550 base pairs, and the first read, which is 300 nucleotides, covers most of the molecule, but not all of it. The second read, also covering 300 nucleotides, and you can see that there is some overlap in between both read pairs, allowing us to associate read 1 and read 2 with the same amplicon that was sequenced. But what happens if we decrease the read length? For instance, if we switch from 300 nucleotide read lengths to 150 nucleotide read lengths, we're still able to sequence part of the amplicon, but you'll note that there's a large region in the middle that's not going to be sequenced, and it'll then be very challenging to correlate read 1 with read 2 to generate a single combined read pair. So when you're doing this, you always want to choose a read length that will completely cover the amplicon that you wish to sequence. Next, we're going to go over a bit of coverage. So in this specific example, you can see that with the target DNA or amplicon, there are two reads forming one read pair. Because there's one read pair for this amplicon, it can be said to have 1x coverage. By adding another read pair, we can get 2x coverage, and a third read pair, we can achieve 3x coverage. In this example, 3 read pairs are required to achieve 3x coverage for one unique amplicon, or one unique sequence. But what happens when you have more than one unique amplicon? In this case, we have amplicon 1 and amplicon 2, each of which we want to have 3x coverage. To achieve 3x coverage, you need 3 read pairs for each amplicon. With 2 amplicons, this would therefore require 6 read pairs, which would give you 3x coverage. Now, often you can have variation in coverage between samples. In an ideal case, if you have 3 reads and 3 unique amplicons, you would ideally get 1 read pair per amplicon. In reality, some samples might receive more reads and some samples might receive fewer reads due to stochastic differences in DNA sequencing. So it's very important to sequence more than the minimum number of reads you may think you need for a project. Next, you need to determine how much coverage you may actually need. The first question you want to ask is how diverse is the sample you're working with? For example, you might have 1,000 unique amplicons that you need to find out the sequence of. If you'd like each amplicon to be sequenced 50 times, or 50x, this would represent 50x coverage. For 1,000 amplicons, 50x coverage would represent 50,000 paired end reads. Next, you need to figure out how many samples you're working with. If you have 10 samples, and you need 50,000 paired end reads for each sample, this would represent a total of 500,000 or 500,000 reads for your Amplicon-Seq project. Next, we'll go over some general guidelines for sequencing depth. For most samples, how deeply you want to sequence depends entirely on your project needs. Different projects may require different amounts of sequencing, such as metagenomic sequencing, where 50,000 reads per sample is quite typical. For antibody screening, you may want as many as 500,000 reads per sample. And for CRISPR validation, you can often start with up to 1 million reads per sample. Next, I'll go over some important considerations for Amplicon-Seq. First, you want to consider how variable your samples may be, or how many unique sequences might exist. We discussed this a bit earlier. Next, you need to ask how deeply you want to sequence the samples. Thirdly, what read length. And finally, what type of analysis you want to do to obtain useful information from your project. We'll discuss this a bit more in detail when we go over post-sequencing analysis, but we'll go over 3 specific examples, including CRISPR screening, antibody screening, and metagenomic studies. Next, we'll go over a bit of the workflow for Amplicon-Seq. There are a couple important concepts to understanding the workflow, but they're very similar to other NGS projects such as RNA-seq and Whole Genome Seq. With amplicon sequencing, the first step is sample preparation, where you take your amplicons and ligate sequencing adapters to them, shown in green and orange. Next, you load your samples onto the sequencer, and a step called cluster generation occurs, followed by sequencing by synthesis, before you can interpret the results from the sequencing run. For all NGS projects, you'd begin with an important step where you assess the input, determine its quality, and proceed to library preparation. For Amplicon-Seq, first you want to ask, is there enough DNA to proceed to library prep? If so, you can generally move forward. If not, you often have to repeat the PCR that generated the amplicon to generate more starting material. Next, you take your DNA sequences and ligate sequencing adapters directly to them. These sequencing adapters are important for binding of the molecule to the flow cell. If the DNA sequence doesn't have this adapter and it doesn't bind to the flow cell, it won't be sequenced. Next is a stage called cluster generation or bridge PCR, where the DNA molecules ligated with the sequencing adapters bind to the sequencing flow cell in step 1. Next, they form a bridge and bind to the other side with the other sequencing adapter to the flow cell. And there's an amplification step that you can see in 3 and 4, where the DNA molecule is replicated until you have a cluster of identical sequences. When the sequencing occurs, the machine will read the DNA sequences added step by step from each cluster and image this to give you the sequencing result. The basic principle of this uses Illumina sequencing by synthesis, where labeled nucleotides are added one at a time to the DNA molecule, and after the addition of each nucleotide, it's imaged to see what fluorescent emission is generated, which can be used to infer the DNA sequence. Quality control is essential at every stage of this process, though, and next we'll go over a couple different methods we use to monitor this. The first is checking the quality and quantity of a sample. You want to determine if the sample may be degraded or low concentration before beginning. In this example here, you can see in the first lane next to the DNA ladder that there is one large band which seems to be at high concentration. In the next lane, it's still one single band, but there's a lower overall amount, which might be a concern if you have a low concentration sample. In the final lane, you can see that there's one smear, indicating that the sample is degraded and you likely don't have a sequenceable amplicon that's present. You can use nanodrop to measure the quantity of a sample. You can also use qubit. With nanodrop, the readings you can achieve are often a little bit inaccurate because it'll measure both rRNA, DNA, as well as salts and contaminants that may be present in the sample. At ABM, we use qubit, which can give us a more accurate reading and help us move forward to the next stage of the sequencing process. Next, there are 2 other tools you can use, the Agilent Bioanalyzer and qPCR at later steps of the process. With the Agilent Bioanalyzer, it can be used to assess the fragment size that's present in your sample. For instance, here you can see on the left-hand side and right-hand side are 2 large peaks. These are used as markers for small fragments on the left and large fragments on the right. Here I've highlighted them in red boxes, and you can see that there's a single peak in the middle, which represents the sample. Ideally, you want your sample to be a single unique peak representing a single amplicon. The size is less important as long as it's one unique peak. By contrast, a bad Agilent result would show you either varying fragment sizes or no uniform peaks, such as in this example here highlighted with a red box. and this would be considered to have failed QC. Next, there's a qPCR step. While qubit can tell you the amount of nucleic acids that are present in a sample, qPCR can tell you how much is sequenceable of the library that you actually have prepared. Next, we'll go over some specific applications for Amplicon-Seq, including CRISPR validation, antibody screening, and metagenomics. So with NGS, the raw output is just the sequencing data itself. This is processed into alphanumeric text called FASTQ, which is a FASTA format with QC info from the sequencing run. Next, this is followed by data analysis, typically where you would align the sequencing data to a reference sequence. But what does the data actually look like for NGS? Typically, you'd see these long strings of letters and numbers, most of which is hard to discern what's going on. But in this red box, I've highlighted the actual DNA sequence from that particular read sequence that is of interest to a researcher. But next you have to ask, how do I actually use this data to do any useful analysis? So we jump back here. The first step would often be to align this to a reference sequence and then work with a bioinformatician to be able to do the analysis that's relevant for your project. With this data, there are a couple of different things that you can do. Depending on your project, you can use this data to determine from a CRISPR study what edits might be present in your cells, with antibody screening to find out what antibody sequences exist, and with metagenomics to determine what types of organisms are present in a given sample. For a CRISPR project, we'll run through a brief example now, but with any CRISPR study, you could use the surveyor method to tell if you have any edits present in your sample. But first, you'd have to generate a monoclonal cell line to do Sanger sequencing and find out what type of edit is present. And you can't use a mixed population of cells or mixed edits. To get to this stage, you would absolutely have to go through generating a monoclone. But with Amplicon-Seq, you can do this entire step with one sequencing run. And it can tell you what proportion of cells in the population are edited, the types of edits that are present, as well as the frequency of edits. In this example, we're going to look at some of the analysis depending on the sample type you're working with. If you have a set of cells that are unedited, and you try to look at insertions or deletions that occurred after doing an Amplicon sequencing run, you'll see that in this example, 100% of the cells are unedited, as shown by an insertion-deletion size of 0 base pairs. In cells where editing has occurred, you would likely see a number of small insertions or deletions, up to about 18 base pairs or so. In this example, you can see that all of the cells have been edited, but the edits are different sizes, ranging from 10 nucleotide deletions as few as 1 or 2 nucleotide deletions. In this third example, where you have a pool of cells, some of which are edited and unedited, you can determine what edits exist in that population. With this, you can determine if it's worthwhile to try to establish monoclones from those samples, rather than wasting months trying to establish a cell line and finding it doesn't contain the edit you're interested in studying. Next is antibody screening. When you're screening for cells that might express an antibody targeting a region of interest, it can often take months to identify cells that express your target. And you'd have to generate a monoclone to do Sanger sequencing to find out what type of target is recognized. With amplicon sequencing, you can use this to tell the types of target sequences that may be present, as well as their frequency. And then you can use this information to predict how likely these antibodies actually are to interact with a given target or antigen. For instance, if you start with a large pool of antibodies raised against one particular antigen, you can use the results from Amplicon sequencing to try to look at the interaction score for different antibodies that are generated in that pool to predict which antibodies may be best suited to interact with a given antigen. And this is a great tool for screening for difficult or challenging antibodies. Finally, we'll go over using Amplicon-Seq for metagenomics. The basic premise for metagenomic Amplicon-Seq is that all cells have ribosomes composed of rRNA. Because rRNA genes are highly conserved, you can sequence these genes to identify specific species in mixed samples and their relative proportion. And then you can use types of analysis or visualizations, such as rarefaction curves or a Corona interactive map to explore your data. For instance, rarefaction curves give you an idea of the efficiency of next-generation sequencing in capturing sample diversity. I won't go over this much here, but effectively, you can use this as a way to determine if the sequencing depth that you chose for your samples was appropriate for capturing most of the diversity that's actually present in the sample. Next, you can use Corona to generate an interactive metagenomic visualization. This effectively looks like a large multicolored pie chart that can give you information for the specific types of bacteria that are present in terms of genus or even species level and their relative proportion. If you zoom in this pink region here, you can see very specific species information, including for Mycobacterium, Rhodococcus, and Nocardia. With all types of Amplicon-Seq, you can do additional analysis, such as single nucleotide polymorphism detection, as well as insertion-deletion calling. For instance, if you go through Amplicon-Seq and get your sequencing data, you can then align the sequencing reads to the reference. If there's a single nucleotide that's different between the reference and your mapped reads, this would represent a single nucleotide polymorphism or a SNP. Alternatively, if your reads map to a region generally, but not one specific nucleotide, such as this G here, this would be considered a deletion that's not present in any of your samples. You could then use this to look at different insertions or deletions that might affect the coding sequence for a given protein, which is important for CRISPR editing. Next, I'd like to hand it back to Nancy to help wrap up our webinar before we go over a few questions that you've submitted.
Thank you, Christopher, for that excellent talk. Before we wrap up, I would like to briefly mention a special promo code that we're offering to everyone who is joining us for the webinar. For 50% off analysis for CRISPR-Cas9, antibody screening, and metagenomic Amplicon-Seq projects to help you take advantage of our dedicated bioinformatics team to help you analyze your data. ABM also offers a number of resources for researchers that are interested in learning more. Our website's knowledge base has many articles covering the wide variety of topics. Our YouTube channels has numerous videos to cover important content in an accessible way. And don't forget to check out our blog for the latest posts covering tools to help you succeed in your experiment. Our technical support and customer service teams can also assist you at every stage of your project with ABM. If you have any question about our materials or services, you can always reach us by email or phone. Finally, stay tuned for our upcoming webinars. Thank you for joining us. If you enjoyed the webinar, please like it and subscribe to our channel to see more. If you have any questions or comments, please feel free to leave them in the comments below. We'll now begin answering your questions. Okay, so it looks like we got a bunch of questions, so let's see. Chris, the first question is, what is the largest size amplicon I can sequence?
That's a great question. So generally for Amplicon-Seq, we ideally want to use 2 × 300 base pair read lengths. With this read length, you can sequence amplicons up to 600 base pairs, although it's ideal to do slightly less, so there's some overlap between the first read and the second read. If you have amplicons that might be larger than 600 base pairs, we can still work with these types of samples, although there's additional steps that are required, and we have to adjust the analysis that's possible as well.
Okay, thank you, Chris. The next question is, for metagenomics, can I do amplicon sequencing for things that are not bacteria, like fungi or plants?
Yeah, so we can do different types of amplicon sequencing for metagenomics, whether you're studying bacteria, fungi, which would have a different target region that's conserved that we'd amplify, or things like eukaryotic species, including plants, where instead of amplifying the 16S subunit of the ribosome, we would do this for the 18S region.
Cool. We hope that answered your question. The next one is, I'm interested in CRISPR validation to look at off-target effects. Can I do Amplicon-Seq for that?
Generally, when you want to look at off-target effects from CRISPR editing, it might be better to use a whole genome sequencing strategy, where you can sequence the entire genome from an unedited cell and compare the edited genome from another sample to look to see if there are any off-target effects. It's a bit more challenging to use Amplicon-Seq for this project.
Okay. The next one is, how long does it take for an amplicon sequencing project? I'm in a rush for my study, and some companies take a few months.
So this depends on what we would begin with for your project. For most Amplicon-Seq projects, it takes about 2 to 4 weeks, including analysis, from the time we receive your samples, including post-sequencing QC, and final delivery of the report. In some cases, we can do it even faster than this, but it depends on the sample number, the read length that you require for your project, and a couple other important details.
Okay, thank you, Chris. Let's see what's next. If there is a minimum number of reads I need for a project, do you guarantee this? Like for 16S sequencing, for example?
Yeah, that's a great question. So often when we're setting up sequencing projects, it's very, very challenging to pick a very specific number of reads and always achieve that. So we will always try to sequence more reads than the minimum numbered for a project. With 16S sequencing, many researchers require about 50,000 reads per sample, and in order to make sure that we achieve this, we will often attempt to sequence about 100,000 reads for each sample.
Cool. I think we are running out of time, so we're going to take the last question, which is, you didn't cover the type of analysis I want for my samples. Do you do any custom analysis? Thank you for your help.
Yeah, so we actually have an in-house bioinformatics team that can help with essentially any type of analysis that you need for your project. We would just need you to give us a bit of detailed information regarding the type of analysis you're interested in and maybe the goal for the analysis you want to do. And then we can work with our bioinformatics team to find the best option for your project and work with you on even discounted bioinformatics pricing that works for your project.
Okay, I think that will be the end of this webinar. I would like to thank Christopher again for joining me for this webinar, and all of you that joined us today. If you still have any questions you may have, then please feel free to email us or leave in the chat box. We will send a copy of the slides and answers to all of your questions shortly. Also, if you are ready to begin with your next generation sequencing project, you can begin by sending an email to Christopher and our NGS team at ngs@abmgo.com. Thank you, everyone.